Well, first of all, of course, many thanks to the organizers for inviting me here. Actually, I do share an over 30 years history with Eugene. No, I'm not that old, but actually Ferdinand had the habit of bringing especially dearest colleagues and friends back home. So I enjoyed very much as a kid his physical presence, which is, I think, impressive for a kid. He brought lots of the treasures of a kid, so the most treasures, namely foreign coins. And he brought this wonderful crowd with him with these fantastic glasses, which me as a child completely impressed me. Just remember of Victor's image of himself and Grecian, which he showed earlier. Unfortunately now, actually, the Soviet ruble as well as the German mark is gone. The glasses, unfortunately, are also basically gone, though I heard there might be some remnants of that. And unfortunately, actually, Eugene's chair is empty today. But what we are left with I'm playing around with is, of course, this huge knowledge and structure about investigation of toxins. And you might remember today's at the opening ceremony, the image where he showed actually the importance of toxins in terms of biochemical studies, and I guess that was the main focus of the little bit advanced aged population in here. So this leaves us now still the cellular, the physiological and the drug aspect to it, and I'd like to show you some examples why actually targeting ion channels might have also a different aspect than you might expect. So my focus of interest is actually pain, the peripheral nervous system. So I guess all of you, of course, are familiar with neuroscience and basic neurosciences. So this is being a peripheral sensory neuron, where you detect all these different kind of physical stimuli. Of course, being a neuron, the most important aspect is the action potential running through the neuron. This is produced by a number of different classes of ion channels. We just heard of the voltage-gated sodium channels, which are, of course, important actually all along the path in initiating the action potential, in transducing it along the neuron, and as well as in the presynaptic activity. The speciality of the nociceptive neuron is actually that it is highly modulated by other substances, by pH, as we heard earlier, but also by ATP growth factors, and you name it. These action actually mostly comes about by non-ionic signaling. I just picked actually as an example two GPCRs, an alpha S coupled to one and an alpha I coupled to one, which among others through the activity of pKa result in this sensitization of the peripheral nervous terminus in an increase in action potential creation, and therefore in more pain. If you now would not know anything about pain, but if you would start knocking out or blocking actually one of these channels, especially the sodium voltage-gated sodium channels, you can easily delineate that this phenotype should be pain-free, because actually at all these levels there's basically no neuronal activity, and that indeed is the case, especially for the NAV 1.7. There are animals, knockout animals of course, but also patients which are known with this mutation, and they are absolutely pain-free all throughout their life. And the logic is so simple: no neuronal activity, therefore no pain. This sounds like a perfect drug target, and the pharma industry has developed a whole range of very specific NAV 1.7 blockers. Unfortunately, though, the phenotype of using these drugs is completely null, so there's no analgesic effect whatsoever. That was somehow disappointing, I guess, at least for the companies. One of the central figures in the field, John Wood, went deeper and had a further analysis of the knockout animals. And what he found is actually an alternative mechanism which is regulated in these animals. Namely, he found a strong upregulation of the mRNA of an endogenous opioid, so of preankephalin in females as well and as in males. But it's not only the mRNA, but he actually also found the protein to be highly increased in the dorsal home. And accordingly, actually, if you now do the animal behavioral experiment, if you take wild-type animals, you could use any kind of opioid blocker, and this, of course, has no effect because there's basically no baseline opioid activity in the normal human, in the normal organism. But if you now use these knockout animals, then the opioid receptor blocker resulted in bringing back the pain-free phenotype to a normal wild-type pain-perceiving phenotype. And that was true for thermal pain as well as for mechanical pain. And more importantly, actually, that study was done on one anecdotal patient, 35 years of age, and also there, so she had no pain perception whatsoever. But if you give an opioid receptor blocker, suddenly for the very first time in her life, she was able to differentiate, in this case, a thermal painful stimulus. So this is completely stunning. It completely puts the whole field upside down. But it has many important studies. It raises a number of questions, namely what really is the mode of action here. So is NAV just only controlling the expression of the opioids, or does it actually modulate the signaling in the cell itself as well? And this, I think, is a very important question, because usually if you take a patient, you just have to show him the opioid and he starts to desensitize immediately. So what really, I mean, a similar mechanism one would expect actually in these animals or in these humans to occur as well. So we were actually collaborating with John Wood and pursued a pretty extensive study which just came out a month ago, and I'd like to share some of the central data there with you. So this is kind of a simplified version of the problem. So we have the knockout of the sodium channel 1.7. What John Wood showed earlier is this upregulation of the proenkephalin and some kind of action of the opioid. Where it's really acting and what that really means was unknown. So we asked the question, is there any kind of change in the activity, in the cellular activity of the mu opioid receptor? Is there potentially actually also the counterplaying parts, so the serotonin induced sensitization pathways, might that also be affected? Both of them actually kind of converge on the regulation of pKa by controlling the adenylyl cyclase. And this is a rather challenging study because, of course, you need primary sensory neurons to be open to any kind of subgroup in the peripheral neuron system to be involved, and you need to basically monitor all of these at the same time on a single cell level. We established a system a couple of years ago being able to do that. It's actually a rather simple approach. We take the primary neurons, we dissociate them, cultivate them in a multi-well place where we perform any kind of stimulation assay. Then we fix the cells, do completely normal immunofluorescent cytochemistry. Then we have a fully automatized microscope, which results in about, actually not 18, but 25,000 images out of one such plate. And there we do software-based object identification and single cell data extraction, which results then in pretty neat multi-dimensional representation of these cells. And you can use that to really calculate those responses and kinetics on a physiologic primary neuron population basis. So we did that. We started out with a pronociceptive GPCR signaling, and as you might appreciate, here on the y-axis is a phosphorylation degree of the regulatory subunit of R2, which we established as an endogenous readout of endogenous pKa activity. So the higher it is, the more pKa activity you have. On the x-axis is a subgroup marker which identifies the nociceptors, so the pain neurons, so the further down here. And this is a dose response with serotonin, and as you might appreciate, it's especially these R2 beta positive ones which then start to actually show an pKa response in the wild type. In the knockout, though, this was still the case, but much reduced, as you might appreciate here, of course, we quantified that, and as you can see, the KD basically does not that does not change at all, but the amplitude does change. So serotonin signaling indeed in these animals is altered, so the pronoceptive input into these neurons is reduced, and this was actually a specific effect for serotonin. We also checked that for prostaglandins like prostaglandin GI2 and non-receptor way of stimulating CMP with forskolin. Also that only showed a little tiny bit of a change, or actually using CMP analogs as well. They basically look the same. So it's very specific, the serotonin input, which is reduced in these animals. But having a look actually at the pKa isoforms in these cells, you might appreciate that these R2 beta, the regulatory subunit, which is predominantly expressed in nociceptive neurons, that this is reduced. It's not that much, but it's very highly significant. So this data is based on quantification of over 100,000 neurons. So the statistic is of course not done with 100,000, but actually with the replicate. So this is a small reduction, but a significant reduction of this sensitizing mediator of pKa. But then of course comes the central question, namely what's about the opioid signaling? And first of all, these are the controls. In red, this is the knockout. In black, it's always the wild type. You might appreciate that the pKa baseline pKa activity is a little bit higher in the wild type than in the knockout. That's what I showed you also in terms of the expression of these pKa subunit there. Fentanyl by itself, an opioid, does not have any kind of effect. Naltrexone, an opioid receptor blocker, also does not have an effect as it should have. Serotonin does result in a nice kinetic as usual, again with this little tiny shift which is caused by the baseline activity there. But then comes the important experiment combining actually the serotonin stimulus with the action of fentanyl. And there you might appreciate that really the activity of fentanyl is much stronger in the knockout animals. And that comes as a very strong surprise, because if you consider that these animals overproduce endogenous opioids, one would expect actually rather a desensitization and not a further sensitization of the system. Of course, this could be just an artifact by an activity on any kind of cell. So we followed up on that, of course, on a more detailed levels. Here is on the x-axis the size of the neurons. The nociceptors are always the small ones, and indeed you might appreciate that this effect is very much specific for the small-sized nociceptive neurons. Again, we quantified that, and again the dose response is basically exactly the same, having the effect in the normal range. But what you see is that actually the reduction of the input actually goes through the whole system, through all concentration ranges up to here. And that is something very interesting, because if you now calculate the efficacy of your drug, so this is basically just the reversal, so dividing one by this number, then this turns out that you have at maximal doses with fentanyl in the wild type only about 60% of reduction of the activity of pKa, while in the knockout you get up to 80% and potentially actually even higher. This is a very interesting aspect. So it's not only that the system reacts more sensitive, but it's actually also a stronger activity of the opioid, which is in these cells. So what we showed in this study is that actually, I skipped the data, but it's really a regulation of the serotonin receptor for mRNA, which is regulated by the presence or the loss of NaV1.7 in the knockout animals. I showed you some of the data showing that actually the serotonin input, the activity really is reduced, therefore actually suggesting less painful stimuli. I showed you that actually also this convergence point, this pKa system, is reduced. this has been shown by John Wood in before, that the PENK gene is upregulated, and we also now showed that this opioid receptor system is increased in its strength. So it's basically 5 different aspects which are all together synergistically regulated, and that's something which came very much to a surprise for us. Very often you see a change in one of these systems, but which is then counterbalanced by the other side. So basically if you put more weight on this side, then the body actually starts putting more weight also on this side. So that's why we opted for this representation. It seems to us that instead of putting more weight on one of the either side, we actually shifted the axis of the seesaw. And you might speculate or think about that this might be one way of regulating actually the homeostasis of this whole system. And what is very interesting is this suggests now that the blocker indeed should not show any kind of analgesic effect, but it should be a wonderful drug to support actually any kind of opioid therapy. And just a few weeks later, actually, there was indeed an animal study showing exactly that. So instead of just looking at the electrical activity, which you might modulate with an ion channel blocker, consider actually also the activity of the ion channel on other systems, for example, like the intracellular signaling. And to further corroborate this aspect a little bit more, I'm very privileged actually to really being allowed to play around with Grishin's treasure of toxins in collaboration with Alexander Wasilewski. So we started out to ask a question, okay, is there actually, can we identify in venoms components which act not only on voltage gated, on ion channels, but actually which might modulate thereby the intracellular signaling. And we went for a number of different creatures, so Alexander knows better than I why we opted for some of them. And I just like to indicate a little bit what we've done there. So the workflow is very simple. Of course, ideally, you look out for components modulating intracellular signaling in an as broad manner as possible, which means screening like crazy. But I learned this important aspect actually from the communication and collaboration here with Moscow, that indeed venoms are the optimal pooling approach, because that pool is really evolutionarily optimized so that you do not have any kind of masking effects or something, but in the best sense actually synergistic efficacy. So we started out with venoms. I showed you this platform which we have. Therefore, we could identify actually venoms which show some activity on this intracellular signaling. Then Alexander performed fractionation attempts. We went back in there to identify really the fractions which have the active toxins, and then processed in analyzing the cellular mechanism. We went quite a bit through this process already. So these are controls: negative control, positive control in terms of pKa activity or ERK activity. And as you might appreciate, we do have some candidates here which indeed induce strong activation of these signaling cascades. We went all the way through the fractionation process and did N-terminal sequencing, and thereby we were able to identify the toxin. I was very much looking out for anything acting on a GPCR or something similar, as my deeply rooted religion here is that ion channels are completely irrelevant. I'm sorry, so I'm the son of somebody who worked on ion channels, so somehow I have to have a very opposite idea here about life. But it turned out to be an alpha toxin acting on a sodium channel, on a voltage-gated sodium channel, and currently we're investigating actually how that really works and why that results in activation of intracellular pain signaling. So what I showed you here now is that indeed, well, this is more a confession I have to make, ion channels are important somehow. I don't know, but might come as a surprise to some of you, but what I hoped that I could convince you is that there's not only this classical activity going from the GPCRs modulating ion channels activity, but actually that we have also quite some activity going the other way around, and that is actually in the pain context something which basically is not explored at all. While we have lots of LTP and studies as such in the central nervous system, this aspect basically is missing. So with that, I'd like to end, highlighting here the people most important for the study, my long-term postdoc, Marianne van Kannen, a very talented student, my collaborator John Wood on the knockout, and then this fantastic group here in Moscow. Many thanks.
Thank you, team. A couple of questions, please.
Congratulations with your talk and the results. As a pharmacologist, I would also be very intrigued to know whether the CB1 receptor, the cannabis receptor type 1, would be involved in, let's say, crosstalking with the knockout, because that's also a very important GPCR involved in pain, it's clearly known. So intuitively, I would not think about involvement of the serotonin, which you clearly demonstrated, but rather go for the CB1. So has this been checked, or can you exclude this?
No, we have not checked, and we cannot exclude that. The study was mostly actually directed by the transcriptome analysis, which showed indeed this increase of the enkephaline, and therefore we looked for opioid receptors. On the other hand, there was some indication of a change of the serotonin system, so that's why we opted for these two components first. But actually, indeed, absolutely, there's a huge number of questions now arising from that. Also, just taking these few components, what happens if we now pharmacologically or genetically target actually the serotonin receptor? Is there a similar counterbalancing effect? So is the whole system really tightly interconnected and always moves into the same direction, or does it not?
You finish with the sacrifice, God blessed words, toxins, but you are focusing on the pain, on the receptors, and what about the species, but you're working with mice. What about the species specificity of these sodium channels? Because there are sad examples where some toxins, fantastically active on mouse receptor, were absolutely dead on human ones.
Right, so quite some of the study actually has been performed in mouse as well as in rat, and so in that respect we're so much closer to the human already. But you're absolutely right. Of course, there's always the doubt in the end of the species difference. We don't know. We're currently working on establishing iPS cell-derived human nociceptive cell lines, which would allow us to analyze that in a more human system. But currently, that's a little bit difficult. At least this core effect of this high efficacy of opioids in humans, that has been shown in this anecdotal case by John Wood.
Thank you. I have a small privilege as a co-chairman. So could we just summarize the message to take home? Signaling by this channel, which you designate as sodium, right? But in fact, the signaling is due to the incomplete selectivity of this channel, right? Or how?
Well, that's a very interesting question. I mean, at the moment, we only have these very harsh...
Not that interesting, but
well, it's
difficult.
Right, we only have this very, very kind of harsh and strong differential situation of either having the channel fully active, so normally active, or completely deleted. So I think further pharmacological studies can address this aspect.
So will you this part of message to take home for the future, right?
Right, so we are actually already in the realm of looking if it's sodium which can affect intracellular signaling, or is it rather calcium?
Which is not totally excluded.
Right, no, absolutely.
Sodium can affect G proteins, right?
That's true.
Okay.
Our collaborator would love to see that, actually.
Thank you again, team. Yeah, all right. Next